Hi Year 12s, this is Mr. Lim here, and this is our last video on proteins about the denaturing of proteins. Okay, so we're going to be learning about denaturing proteins. Okay, so what is denaturing proteins? So remember, the protein's function is closely linked to its shape. So the shape of the protein is maintained by the many small interactions at the secondary and tertiary level within a protein, and if it's a complex protein, at the quaternary level as well. Okay, so the shape is held together by many small interactions. If a significant number of those interactions are broken, the protein will unfold or turn into a different shape, and the process is called denaturing. Okay, so denaturing can occur if the proteins are subject to change in the environment, mainly in temperature and pH. Right, so most organisms will then try to maintain a level of homeostasis or constant internal environment to ensure that the conditions don't change too much so that the proteins stay functional and the chemical reactions can still continue to occur at biological temperatures. So here's my protein. Okay, it's a tangled mess of stuff, but it's in that shape, right? It's being held together by all those bonds or all those interactions. Okay, it's held together. If you break enough of those interactions, I didn't mean to do that. But if you break enough of those interactions, then the protein turns into a whole brand new shape, and then it's like, oh, it's not supposed to do what it's supposed to do, and therefore it's denatured, right? So we can do it, denature a couple of ways, by temperature or pH. So let's talk about temperature. Increasing the temperature increases the average kinetic energy of the particles, which means that they vibrate a bit more. When the particles have more energy, they can break free of bonds that hold them together, just like they're boiling, right? So if the temperature of the protein is increased, some of those small interactions at the secondary and tertiary level will break. So here's, that bond that's holding those things together, right? If these parts vibrate enough because they've got so much energy, then that bond is broken. Okay, and if that bond is broken, then it's likely that this thing just turns into some wider shape, and then it's no longer in the original shape that it's supposed to be in, and then it's denatured. If a significant number of interactions holding the proteins together break, because you have to break quite a few of them, then the protein will unfold and form a new shape. Okay, so if the temperature does not return to the original level, so even if the temperature does return to the original level, the proteins will still hold its new shape, because the folding of the protein requires other factors which occur immediately after a protein is produced. So remember when we talked about primary structure and how we said the amino acids are kind of gathered from the cell, and then they're just kind of added one by one by one by one by one. Right immediately after that protein is entirely built, it goes somewhere to be folded by other proteins into the right shape. Okay, and so since when you denature something, maybe those proteins aren't there, or maybe they've been broken as well because of the temperature, then they're not going to go back into the right shape because those other proteins aren't there to do the folding, right? And then an example of that is like when you cook meat, effectively what you're doing is you're taking all those proteins within those muscles, and they're in a certain configuration and they look a certain way. When you heat them up, you're breaking all of those proteins along, and even those proteins, they're actually sitting in like nice discrete packets. So that's one protein, that's another protein, and that's another protein. Okay, when you denature them, right, they start to untangle, and what they'll do is that they'll actually tangle with each other. So that's just one of them, that's another one of them, and that's another one, and then they'll tangle up with each other, and they won't ever go back to their original form because they're all tangled up and they're all terrible. All right. You can also denature proteins by pH. Okay, changes to pH changes the nature of certain side groups of amino acids. So just like Zwitter ions, how their carboxylic acid end and the amine end will change the way they looked at different pHs, what will happen is that side groups will also change how they look at different pHs. If you've got a carboxylic acid end of a amino acid on your side, or a carboxylic acid on your side group, if you change the pH, that can go to a COO minus or a COOH, and therefore you may, if it goes from that to that, you may not be able to create an ionic interaction. You may not be able to hold things together, and therefore everything falls apart. All right, so some will lose the ability to form ionic interactions, while others might gain the ability to form ionic interactions, and then all of a sudden you're joining to places where you're not supposed to, and then it's all wrong. If a significant number of interactions break, then it unfolds, forms a new shape. Like temperature, the change in shape due to pH is permanent, and therefore proteins will not return to their original shape after pH returns, and that's why it's so important to keep the pH inside your body, and why acids are bad, bases are bad. All right, so that's the denaturing of proteins, and that's it for proteins. Make sure you go do your assignment and work hard. Adios.